Well, I'd like to begin by thanking the organizers for the opportunity to speak today, and thanks for all of you who braved Salesforce to make it here this morning. So I know that this was supposed to be sort of talking beyond the cell atlas, but I wanted to start by saying that we're very much in the cell atlas phase right now. In particular, the brain efforts have been really well initiated and funded. We're actually part of a number of different efforts now, trying to plumb the diversity of cell types in the brain, which I was sort of struck from a set of conversations from a Stanford meeting a couple of years ago, where the different organ groups were actually asked to what extent they thought that they were going to learn many, many new things from single cell work. And most of the organ people thought that they probably already knew 90 or 95% about their organ, and they're going to discover a little bit more. The brain people felt that it's 95% discovery. And I think really that's true, and that's particularly true for the human brain, where we simply haven't had access to this sort of very detailed cellular resolution. And the transcriptomic approaches have really cracked open this field in a big way, that suddenly we have access to this wealth of different cell types that make up the nervous system, and the genes that give rise to those properties. So just to sort of call out some of these things, we've been working on this at the Allen Institute. The Brain Initiative has started this cell census network, which is really pushing towards whole brain in the mouse and eventually in the human, and the Human Cell Atlas. And I especially want to call out in the context of this meeting what I think are really the great contributions to this field from the Chan Zuckerberg Initiative, both through the BioHub, but also through supporting Human Cell Atlas efforts that we're part of, and also in terms of sort of open science, but also team science and consortia. We're part of a consortium to try to benchmark and optimize spatial transcriptomics methods, which is really an unfundable type of thing through traditional mechanisms. So I think it's really a great contribution to the field that CZI is making. I'm going to spend most of this talk on one particular topic instead of sort of a broad overview. The advent of being able to use these transcriptomic approaches in human has really, I think, for the first time now allowed us to take a quantitative approach to understanding specialization of the human brain. So I'm going to sort of feed off of Detlef's talk last time, which is about conservation even to the earliest animals, to instead talk about specialization. And what is actually distinct about the human brain? Is it just a larger version of a mouse brain, for example, that we like to use as a model organism? Or is there something qualitatively different about it? Of course, we know certain things about this, that we have differential expansion of certain regions like the neocortex, and even differential expansion of certain parts of the cortex. But we really don't have a good understanding of how well conserved this is. And of course, this has clinical relevance as well, because as all of you know, most drug studies that use mouse as a preclinical model organism don't actually make it into the clinic. So I borrowed this from a cancer thing, but this could be almost any field in biology now, that there's an element of being sort of lost in translation, moving from one to the other. So maybe this is a way to help to begin to understand that as well. So the approach that we've taken at the Allen Institute for some time is to really go for deep coverage and try to understand a very fine diversity of cell types in the cortex in particular. This is from the initial study from my colleagues, Basilika Tasic and Honghui Zhang at the Institute, where they tried to use a series of mouse Cree lines to isolate various populations of cortical cells, put them together, and do a clustering to derive a classification of types. And you see many versions of this. I prefer the hierarchical tree to the T-SNE sort of diagram, because it has clearly sort of lays out the organization of the different major types of cells that are differentiated from one another based on transcriptomics. And this can be taken down several additional levels that really mirror the developmental origin of different kinds of cells. So suddenly, we sort of have a quantitative approach to understanding the diversity of types and the genes that differentiate them. Now, when we first did this study, we had no idea how well saturated this was. Have we actually covered most of this? This could be a highly biased population. And so Honghui's group has done a much denser coverage now. This is available on BioArchive, which led to a couple of interesting principles, I think. So the first is that transcriptomic diversity is not infinite. In doing about 10 times the number of cells, you only see about twice the number of cell types. In fact, this is really saturated for most of this. So there is kind of a finite answer that one gets that's quite complicated, but far more manageable than it could have been. So for example, within any given region of the cortex, we can differentiate about 100 different types of cells. This represents both neuronal and non-neuronal types of cells. I should note that this row is showing the numbers of cells that are in each of these clusters, which I'm going to contrast a little bit later, is not an accurate representation. This is a consequence of cell dissociations, having biases associated with them, and the use of Cree lines to try to cover populations which are more difficult to cover. But also, again, to highlight the sort of structure of this, it's very much sort of what Detlef was showing yesterday. We have these big clades that clearly sort of are related to developmental origin. And in fact, there even are a couple of outlier groups that behave in a certain way that are quite different from others. And in fact, they come from different developmental origins as well. So the cortex is actually sort of a mixture of cells that are generated locally and coming from distant regions. One other sort of principle that comes from this is that the GABAergic cells, the inhibitory cells, are actually common across different regions of the cortex, whereas the excitatory cells are different enough that they cluster separately in different regions of the cortex, which is, again, sort of consistent with the inhibitory cells coming from an external source and coming to populate the cortex as a whole. So this is very nice in the mouse. It caused a problem with trying to translate this to human, namely that it's not really possible to isolate intact cells in the adult human brain, even from neurosurgical resections. So this forced us to go to the use of nuclei. Lots of people are using nuclei now. When we started this was not at all the case. And we've done a number of careful studies now to demonstrate that you can get a very similar result using nuclei in whole cells. This is a nice study that's on BioArchive now, comparing whole cells and nuclei from mouse cortex, where we could get very matched populations. And we can get very, very similar clustering results. Marker genes show very similar profiles. And really, the one trick is that most of the signal is in the introns. So you have to be using intronic sequence from the nuclei to get this type of result. But this is important for what I'm going to talk about coming up, of comparing mouse versus human, that we are able to achieve similar results with those two different types of data. So what we've been working on for the last few years now is to build a human taxonomy. We've decided to tackle this in the middle temporal gyrus of the human cortex. The reason for this is, I won't talk about much in this talk, but it's because we can routinely get surgical access to those tissues through epilepsy surgeries, which allows us to actually start to layer on other properties of these cells connecting to the transcriptomics. So there is a reason we picked that particular area compared it to other areas as well. And so the idea is, we don't have the ability to select different cell populations like we do in mouse, and so you just have to run the numbers. So we picked a target for the frequency of cells that we were trying to get, and we ran about 16,000 nuclei that were highly biased for neurons to capture neuronal diversity. Another important thing for human is that there's a wide range of different circumstances that you get cases from. So you get post-mortem cases that can vary by the cause of death, by the agonal state, by the post-mortem interval, or surgical specimens, which came from someone who had to have a part of their brain removed. And so part of the study was actually to compare if we got similar results using different types of specimens. And the very positive news for the use of this technique is that cells from even acute surgical specimens and post-mortem specimens out to about 24 hours cluster together. This is not to say that they don't have a signature associated with this, but they cluster together, so we get the same basic results with these different types of tissues. We do, however, see signatures of tissue source. So for example, the post-mortem signature has a certain set of genes that are upregulated, including ribosomal proteins. The neurosurgical signature has actually a whole set of activity-dependent genes that are perhaps reflective of the sort of acute injury of having removed a piece of tissue, the stimulus that caused these genes to go up acutely. But the point I'm trying to make here is that these cell types are quite robust to the tissue source. And so to make a long story short, putting all of these data together, we can achieve a classification that looks quite similar to what we see in mouse using these human tissues. It's on order about 80 types of cells that we can discriminate within this one region of cortex. And as I'll come back to later, some of the reasons that this is a smaller number of types compared to the mouse is that the non-neuronals are really underrepresented here. And as we've started to add more, we start to pick more of these up. So actually, the result is quite similar between mouse and human. One thing I wanted to point out as well is that this is a really interesting feature of the organization that we're seeing, which is that nuclei tend to give a much more accurate representation of the true proportions of cells. We've done a lot of validation with in situ hybridization. So unlike the cells, the nuclei seem to give better proportionality. And one of the things that we see is that basically everything is rare. At this sort of level, especially among the interneurons, the cortex is populated by a large set of types that are all rare. This is not true for the excitatory cells, where some of the populations are really sort of dominant populations. But even there are many types that are in very small proportions. So this is actually quite like the retina in that respect, that there are many types of cells in very small frequencies, which would suggest that they may have particular roles that they're trying to serve to make the cortex work as a whole. A few additional little details that have come out of this that I think are interesting. One is that if we look at the most selective genes for these different types of clusters, almost half of them are non-coding RNAs. So actually, many of the non-codings are far more specific than the coding RNAs, which I'm not quite sure what this actually means. This could be partially a consequence of using nuclear sequence, but it's a striking finding that as we've gone to sort of name these clusters, many of them are actually named by these genes that are along non-coding RNAs or various other types of non-coding elements. A second interesting feature from just trying to make sense of this data is that we approached this by doing laminar dissections, and the laminar dissections are not perfect, but they had predicted that there would be, these cell types actually span multiple layers. This is not expected for excitatory types of cells, which are typically referred to by the layer in which they're found, but it turns out to be true. So first of all, cell types are now defined by combinations of genes, so it takes multiplex FISH to actually go after identifying these in tissues. And when we look at many of these, this is sort of best seen perhaps in the schematic down here, many of these excitatory cell types actually span multiple layers. So we don't know yet whether this is something unusual about human cortex or a human association cortex, but it's an interesting sort of new perspective that these molecular types don't obey laminar boundaries in quite the same way. So now I'm going to move to the actual species comparison. And very much sort of like Detlef showed, I think, for the different species, the overall architecture of the taxonomies between mouse and human turn out to be quite similar. You can see the big groupings of glutamatergic, GABAergic, the different ganglionic eminence subdivision-derived cell types. All these are quite well conserved up to that sort of class level. And now we want to try to quantitatively approach how to match these things at a type level. I want to head off some of the potential critique right out of the gate. There are many reasons that this might not work that well. We're trying to compare cells versus nuclei. I tried already to convince you that using mouse that this didn't seem to be a big issue. We're comparing young mice to much older individuals. Other transcriptomic studies in the past have shown that expression is quite stable across postnatal development into aging. We're using acute mouse versus human postmortem, but hopefully that's not such an issue as I described earlier. The biggest one is probably we're not comparing the same part of the cortex, or this is human MTG versus mouse V1. But at least for the inhibitory neurons, we saw in the mouse that these are quite similar across regions. And then the sampling differences I mentioned before, but that doesn't affect the typology too much. So to do this, we had to employ some techniques that looked for shared conservation across species. If you just do PCA analysis, you get a perfect split by species. But if you use techniques like CCA, then you can start to bring these much closer in register to start to compare between the species. And so the basic results of this are that the conservation across species is actually quite good. With some of the types, we can actually line up one-to-one. Very distinctive types like the chandelier cell, for example, are one-to-one match across species. Others are small numbers to small numbers, but there you can see most things are along the diagonal here. A few things, there seem to be relative expansions in the mouse. This is probably not really accurate because we can't sample the GABAergic cells as well, so we have sort of a trouble getting sparse populations. And as we're getting more data, we're trying to see whether these start to collapse as well. But the basic result is that things match reasonably well. The excitatory cells, which I've already talked about vary somewhat between areas, are actually a bit less conserved. And there are some interesting elements to this. There's one set that are in the layer 4 through the supragranular layers where we actually see a human expansion. And this very likely corresponds to this expansion of supragranular layers in evolution. We see much less diversity of subcortically projecting pyramidal tract cells, which I'll describe more in a moment. And we also see an increased diversity of corticothalamic cells in mouse compared to human. But that's probably due to the region that we're looking at, because there's much more thalamic feedback in primary sensory areas than you would expect to have in an association cortex. But the sort of gestalt is that we're able to line up at about half the resolution in either species between species. And so we can come up with a consensus classification where it turns out that about 10 different types are significantly conserved, that it looks like it's a one-to-one match between species. And most of the rest are collapsing at one node higher than the finest tree. So really very much like Detlef showed us in these very early organisms where evolution's acting at these fine little types. We're not getting major new branches, but we're getting variation at the terminal leaves. And this is extremely good news for studying human because it's so difficult to get other pieces of information about these cell types. This means we can begin to infer information from the mouse to understand the human. So for example, there are many, many of the mouse types where we're starting to get information about what kind of cell they are, whether this is a neurogliaform cell, or a single bouquet cell, or bipolar cell, or a basket cell, or Martinotti cell. That level is conserved, and so we can begin to transfer over these sort of cardinal features to understand what it is that we're looking at. And this is really important because some of these features are completely inaccessible in humans still, even with current techniques such as developmental origin or long-range projection targets of different kinds of cells. Okay, on the other hand, we also see lots of differences. We're not the same as a mouse. And these range through a variety of different categories. One category is that there are different proportions of cell types. I'll describe a few examples of that. There's highly differential gene regulation even within these transcriptomically conserved types. There can be extremely high phenotypic variation in conserved types as well. And there also seem to be some highly specialized types that I won't describe today. So the first is we started now to examine whether there are sort of systematic differences now that we have markers in both types, and we can go back in situ hybridization and other techniques to look at these in tissues. And a lot of this is sort of building on things that have been described before. So there's a very nice study from Arnold Kriegstein's lab recently looking at development of inhibitory cells in human. And using a marker-based approach, they had predicted that there's a much greater proportion of caudal ganglionic eminence-derived interneurons in human than there is in mouse, and a concordant decrease in the MGE-derived populations. And in fact, we can confirm this now, that there's probably a 25% increase in the CGE-derived cells in human compared to mouse, and a drop in the MGE-derived populations. This set of markers we use actually happen to selectively label in both species also one particular type of cell, which is a deep layer neurogliaform type in the mouse that shows a much bigger difference, like an 8-fold difference, particularly in the deep layers of cortex. So I think it's very likely that as we start to look at all of these types to get the real spatial census, we're going to see that there are lots of changes in the proportions of these very specific types of cells between mouse and human. An even more dramatic example, I think, can be explained by evolutionary scaling principles. So as I mentioned at the very outset, the human cortex has expanded dramatically compared to the mouse, and disproportionately. So in fact, it's expanded about 1,200-fold compared to the mouse, but its subcortical targets have only expanded by about 60-fold. So there's a 20-fold difference in the source and their targets that would be impossible to maintain, I think, to have. And so there must be a decrease in the cells that are projecting to those targets to match that. And in fact, that's exactly what we see when we look at these pyramidal tract types of cells. We see that compared to the mouse, where that type of cell represents a fairly sizable portion of the layer 5 pyramidal neurons, they're extremely sparse in the human. And that turns out to be true across multiple areas. And in fact, it's about a 20-fold decrease in the proportion compared to mouse. So it seems that the evolutionary solution to this differential scaling has been to sparsify the populations of deep projecting layer 5 neurons. But these cells also have local connections as well, so this is going to have a significant impact on the structure of the local circuit. So then, now that we've matched up these cell populations, we can also look at differential gene expression for any given type of cell. And here there are some interesting results. First of all, about 10 to 20% of all genes show a very large difference between species, of more than a 10-fold expression level between species. So 1/5 of the genome for any given cell is highly differentially regulated between mouse and human. This is almost certain to lead to some type of phenotypic differences within these cells. The other sort of surprise, although this has been described several times before, is that non-neuronal cells are actually less well conserved than neuronal cells. So you might have expected the opposite, but it turns out that many more genes are differentially expressed between species in the various non-neuronal populations. One particularly vexing thing that comes up is that many of the marker genes that we use to define cell types are not the same between species. So in fact, it turns out that the more selective you are, the more likely you are to change between species. So these highly differential genes are actually more likely to diverge. Finally, I just wanted to make a point that if we look at the gene ontology of these differences, many of you may be thinking, well, is this actually meaningful? The gene ontology predicts that these will be highly meaningful. You have categories associated with neurotransmission, with neuronal connectivity. The top category is actually serotonin signaling. So these are likely to be very functionally important. The last point, if I could have just 2 more minutes, is that I want to make a point about specialization versus conservation. So it turns out that a number of these transcriptomically conserved types actually have very divergent features. A particularly nice example of this is a type of astrocyte, which is found up in layer 1 of the cortex that has a long process. These are GFAP-labeled processes. It can be up to 1 millimeter long in human and in primates, other primates as well. They're called the interlaminar astrocytes. This is not seen in a mouse, where they are much more sort of stellate, protoplasmic types of astrocytes. And it turns out that we see 2 types of astrocytes in our transcriptome data, one of which is enriched in layer 1, which we've now confirmed is that interlaminar astrocyte. But I wanted to show this particularly since Sten is here. It turns out that Sten has also described that there are 2 types of astrocytes in the mouse cortex, one of which is enriched in layer 1, but doesn't look dramatically different from the others. So they have a conserved type, but the phenotypes become very, very different. They're 10 times larger. They have a millimeter-long process in human. They have lots of differences in gene expression, and yet from a transcriptome point of view, this would be considered a conserved type. So it's a very interesting sort of semi-oxymoronic sort of statement that we have conserved types that are highly specialized. So that's sort of what I wanted to tell you about today. I think this is pretty exciting times because it's the first time we can actually really do these comparisons and start to explore this space. And the story is a very interesting one of sort of overall conservation, but then a lot of specialization. And I would like to leave you with the point that details matter for these sorts of things. And if we really want to understand human and we really want to understand how to treat human disease, we have to study human in addition to model organisms. And finally, I'd just like to thank, there are many, many people at the Allen Institute involved in these projects. We have very much a team environment. There are a few people that have been particularly instrumental in the projects that I talked about today. But actually, it's part of a much larger ecosystem at the Institute to work on these problems. Thank you.